Hello everybody, today we're going to talk about a clade called the Ecdysozoans. Ecdysozoans are eumetazoans with bilateral symmetry and triploblastic, and they are two phyla that we're going to talk about today: the nematodes, Nematoda, which are the round worms, and also the arthropods, which are most common ones you know of are insects, but it's an incredibly diverse phylum. And we'll see a lot of different examples or different kinds of groups that are within the arthropods. This ecdysis is a characteristic that's shared between these two phyla, and that is the necessity of molting or shedding the outer body covering, which serves as a skeleton, in order to grow. So they'll be the same size for a little while, and then they'll molt and grow at the same time. They'll be the same size for a while. So what this phenomenon looks like in our example of terrestrial isopods is that we see in this top left-hand corner, this individual has just shed the front half of their exoskeleton, and the new exoskeleton here looks a little bit different. So when we look at that, it's like a perfect little outline, but it's translucent because it doesn't have the body pigment of the body that would normally be inside of it. And this is an unusual molting pattern for terrestrial isopods. They shed the first half, and then in a couple days, they shed the back half. So they molt in two parts. Most arthropods are going to do their molt of the whole exoskeleton all at once, but that's what it looks like. So this molted, cast off molt is like a mold. It's like a perfect little model of what the organism just finished looking like. So it's more like a suit of armor that's a knight can take it off and put it back on again. Now these animals can't usually take it off just when they're doing this process called ecdysis. And it is going to happen at the control of a variety of hormones. So we're looking at these phyla. We're going to spend quite a bit of time on the arthropods because they're the most diverse phylum on the planet. And when I say that, and when you look at this figure, hopefully you might be noticing that perhaps the diversity, the number of species in each of these phyla, is being shown in this diagram by how big the animal is. So obviously this fly is gigantic and out of proportion to that tiny little deer that's right under its foot. But that's showing the relative number of species that we find in each of these taxa. So that deer is representing vertebrates, and we have a comparatively little diversity, about as much as jellyfish, according to this diagram. So the other phylum we're looking at today is represented by this cartoon here, this roundworm. And they're pretty diverse, actually, although it's hard to tell them apart from one another when we look at individuals, and we'll look at their characteristics in a minute. So the nematodes are called roundworms because their bodies are actually round when we take a cross section. So the other thing to notice about their bodies is that they are long and skinny, worm shaped. They're a little bit pointier at one end, a little bit rounder at the other. Their body surface is very smooth. If we take a cross section through the body, it will have a perfectly round shape, which is how they get their name, the round worms. They're not flat worms. Their body isn't flattened. They're not segmented worms. You do not see external body segments on them. They are round worms, and the name of the phylum is the nematodes. So when we look at their bodies, the small ones are mostly translucent, and we can see that most of the body is taken up by these reproductive organs, and that about half the nematodes are parasitic, about half are free living. They're incredibly common in the environment. Many are microscopic though, so you won't have seen them. We're going to look a little bit more at this idea of what the cuticle is and how it is molted in order to grow. So this outside body coating that functions as an exoskeleton, although it's not as rigid as the exoskeleton of the arthropods, it is a protein structure that's stiff enough that it gives the worm an outer coating that the muscles can flex against, and it's going to support the body. So the cuticle is non-cellular. It's molecules that are secreted onto the outside. And we can see in this diagram that cells are underneath layers, the muscle fibers, and the hypodermis, the outer body coating, are on the inside of the cuticle, and the cuticle itself is on the outside. So it's translucent. When we see a nematode worm that's going through a molt, it'll be slipping outside of that cuticle. It'll look like it's coming from outside a plastic bag or something. So down here in this figure on the bottom left, we can see as it's sliding out of its old cuticle, it's going to be able to grow a new one. Now, these animals only grow when they're a juvenile. Once they get to full adult size, they're going to stay there. They aren't going to grow. And they're going to go through 4 larval stages with a molt in between each one. So we can see that on this lower right-hand graph that the size of the animal is going to stay the same until it goes through the molt. So as we're looking over time, we don't see the size increasing directly, that we have a jump. We have a specific time when a molt is happening, that the size is going to increase rapidly during that short period of time, and it's going to go from one stage until the next stage. So J1 to J2, for example, is going to happen when it goes through this molt. So eventually it does its 4 molts, it gets to its adult size, and then it stays there. We look at the body of the male and the female nematode worm. They have separate sexes, so we're always going to see dioecious individuals, and that when we look at the tip of the tail, we can tell a female apart from the male, because the male is going to have this little curly Q-shaped tail, and the female's is going to be straight. The main body feature we're going to see, like I said before, is the reproductive organs. They're going to fill up most of the internal body, but they are going to have a complete gut. It goes all the way through the body. They are going to do respiration through their skin. They don't have a special respiratory organ besides their skin. They have a nerve ring and nerve cords. They have 4. They have a special excretory structure that is called a Renet's gland. So they do have an excretory organ. And they have, let's see, rudimentary sense organs near their anterior end. Basically, when we do a dissection of these worms, which you're going to see in lab this week, is that they have this body space inside that's mostly filled up with reproductive organs. So they do have a body space, but it's only lined on one side by muscle tissue. So we have a pseudoxylem. We do have a cross-section slide of the body of a nematode in lab activities this week. Make sure you look at it, you find the muscle tissue, and you can see where it is. It's only on the inside body wall. It isn't wrapped tightly around the gut like we see in the earthworm. All right. They're able to do locomotion by wiggling. They have longitudinal muscles, but no circular muscles. So their cuticle acts as the circular reinforcing structure, and so their diameter stays the same all the time. These longitudinal muscles, or ones that go along their body, as they flex back and forth, they're just going to make the body wiggle. So when we see a live nematode, it's actually very easy to tell that we're looking at a worm that's in this phylum, because we see this very squiggly, snake-like, S-shaped body movement. So if you're looking at a video of a live nematode, or you do see an actual, maybe a nematode that's in pond water, you see this a very characteristic body movement, and that is because the structure in its body, it only has longitudinal muscle that only allows it to flex back and forth. So it doesn't do it much good if it's in the water, it just kind of flops back and forth. But if it's in soil or if it's in another solid structure, just like a snake squiggling along, it is able to get traction inside that structure and it is able to move forward. Going through log drench to make sure I got everything. LOGR DR. Oh, circulatory system. They just use their pseudocelome is their circulatory system. So there's this fluid spilled space, and as it sloshes around, the largest of our nematode worms is actually the kind you're going to see in our dissection, which are sort of scary, large enough when you think they're a parasite that's going to be inside somebody's body. But many of the species are microscopic or barely visible to the naked eye. All right. When we're looking at different species of nematodes, because their external characteristics are so similar, they actually have to be dissected and you have to look at the reproductive organs in order to tell the difference between them. So it's very difficult to tell the difference between these species, and more or less they all look exactly the same. They're just bigger or smaller on the outside. All right, moving on to arthropods, our second phylum for the day. So they're still an ecdysozoan. However, their exoskeleton is reinforced, their cuticle is reinforced with chitin. So that applies to all the different taxonomy groups. In crustaceans, which are marine and freshwater, the cuticle may be further reinforced with calcium carbonate. So when we think about how hard and crunchy the outside of a lobster or a crab is, especially a large one, they don't just have chitin, which is actually fairly flexible. They also have calcium carbonate. So we might see in crustaceans. Right. That's useful for them because it allows them to be out and about and have an even harder exoskeleton to protect them from damage. It's easy to get the calcium from the seawater, and also because they're marine, the weight of the exoskeleton isn't so much of a problem because they're partly buoyant from the water. All right, so they have this exoskeleton. Now, if the exoskeleton were just uniform and stiff, they wouldn't be able to move. So they have to have jointed regions, like if you imagine how you build a suit of armor for a knight, it has a shoulder cap, and then there's this next section is fastened underneath with some kind of joint. They have to have leather straps that would allow the pieces which overlap one another to still move. So we see that the exoskeleton is jointed. So it has thinner places where it's more flexible. And then we also see that we have a typical structure is to have one pair of legs or some kind of appendage on the different body segments. So another characteristic of the arthropods that's different from the nematodes is that although they have this exoskeleton, their body is segmented. We'll be able to see those body segments and the importance of what things are where on which body segments as we go through these different groups. So we're doing general arthropod characteristics. So we're going to run through log drench characteristics for arthropods to start off here. Their basic body plan is going to be to have this exoskeleton so their locomotion and skeletal support. They have muscle bundles that are fastened to that exoskeleton on the inside. So for skeletal support, they have exoskeleton. It's not an endoskeleton like ours is on the inside, it's on the very outside of their bodies. For osmoregulation and excretion, they do have a body organ, right? So it's going to be different in different taxonomic groups, but the name of it. So we'll start by just saying that for osmoregulation, and excretion, they do have a body organ. They aren't doing it just with diffusion. It has a different name in all the different groups, and so we aren't going to dwell on what it's called. I'll give you an example for the insects later on. For gas exchange, we're seeing that when we have a terrestrial arthropod, it's going to have structures called trachea. And these trachea are tubes, extensions of the exoskeleton that penetrate inside the body and branch until they get tinier and skinnier. And they actually form a pathway like the pipes that bring air into a large building. They bring air inside the body so that oxygen can be diffused to the cells pretty directly. There's also a fluid-filled space in here in the body, so the gas exchange goes along with circulation in order to deliver oxygen to the body. Now, if we have a marine or aquatic organism, instead of trachea, we're going to see gills. And the gills are going to be high surface area that are exposed to the water. They're going to do their gas exchange that way. So if it's terrestrial, it has trachea. If it's aquatic or marine, it has gills. For digestion, they have a complete gut. It has some accessory organs as well, but the basic, so there's a little shadow of the gut that's shown here on this grasshopper. So they have a one-way complete gut. It has some digestive glands hanging off to the side, which is where the food digestion and absorption is going to happen. For reproduction, they have sexual reproduction, and most arthropods have separate sexes. For identification, you're going to look for something that looks like it has the exoskeleton. Sometimes that's hard to see. So legs is the thing that's the most obvious. Does it have legs? If it has legs and it has more than 4 or 2, it's an arthropod. And as you have some experience looking at different examples, you'll be able to tell the difference between those big bristles that we saw on the polychaete worms in the annelid phylum and the legs of an arthropod, right? So the legs may look different than one another. So if they have some kind of appendages sticking off of them and some of them look different, like we have these big ones here versus these little ones for this lobster, then those are legs. They're not just bristles or hairs. The nervous system is shown in this figure up here, and they have a fairly sophisticated nervous system, a brain and a ventral nerve cord, and they also have segmental ganglia, just like the annelids did. And so they have a big enough brain that we think about insects and crustaceans, that they are capable of some kinds of fairly complicated behavior, including social behavior. For the circulatory system, they have an open circulatory system. They have a dorsal pumping vessel. And a way to think about that is it is like a long, large blood vessel. And instead of having one place in it where we have a big ball of muscle that we're going to call a heart that does the pumping, we have a long, large blood vessel, and it's going to be capable of contracting all along its length. Right? So it's going to contract, it's going to sort of squish body fluid out into this body space, and then when it relaxes, it's going to suck body fluid back up into that long body, that long dorsal vessel. So this is called an open circulatory system. That means that it has just open-ended tubes. This is a contrast to the closed circulatory system that we have ourselves, where we have this fluid called blood, and it stays inside of our blood vessels all the time unless some tragic accident has happened and it's squirting out. That's just not how it's supposed to. In an open circulatory system, there is some inside body space that's filled with fluid. Some of it gets sucked into this tube, and then it sort of gets squished around, and then it gets mixed again. So it's less efficient in molecules around a closed loop like a closed circulatory system does. So when we looked at the mollusks, we had most of the mollusks are slow, fairly sedentary organisms that have an open circulatory system. But when we get to the cephalopods, they're large, active, fast-moving predators. They have a closed circulatory system. So it goes along with having a much higher metabolic inactivity rate. One of the reasons why we don't have giant arthropods that have taken over the world is because they have this open circulatory system that is less efficient. So in a lower oxygen environment, maintaining a very high metabolic rate for a long time is not possible for them. All right, so we're going to look at mostly the diversity of different arthropod groups. And we are pretty sure that the ancestor of all the arthropods was a critter that looked more or less like this guy. And what we notice is that it has the exoskeleton, it has the body segmentation, it has one pair of legs on every body segment, and it has these large, obvious eyes. So this characteristic is unique to the arthropods, the compound eye. It's called compound because it's made of many substructures that are all the same, and they're called ommatidia. So this compound eye is going to be capable of forming images, much like our eye is, although it forms them in a somewhat different way. The way the image comes together is different. And we can have different groups that have very large, complicated compound eyes with hundreds of ommatidia, and we also have organisms that have only a handful of ommatidia in their eye. They may also have a simple eye or an ocellus or ocelli, which is a simple eye that just detects light dark. They may have more than one of those. They may have pairs. They may have ones in different places of their body. The other thing that they may have, and it's not shown on this little guy, is antennae. Antennae are another sensory organ. And they stick off the front of the head and they can wiggle them around, and they're used for both mechanical senses and chemical senses. So these are the basic, this is the basic plan of the arthropod body. And we'll see that as it evolved and diversified, remember we talked about in annelids how they all have this body segmentation, but all the segments are the same, and all earthworms pretty much look the same. But they have segmentation. They didn't use it in order to diversify into all different kinds. The arthropods used their body segmentation to turn into many, many different kinds. And so we're going to look at how they have their body segments. Are they mostly the same? Or do they have a sort of different parts of the body that have different kinds of appendages? And then also what appendages do they have? So here are some examples of different groups that we're going to talk about. We have some things like millipedes, we have trilobites, we have spiders, we have some kind of shrimp or insects. So there's a great variety of different taxa, and we're going to talk about those. So when we're looking at so many, many different kinds of species, there's been some variety of kind of reorganization of these phylogenetic tree diagrams. And where we're at right now is the arthropods have two major groupings. One of them is this one called the Chelicerata. The Chelicerata, so chela are pinchers. So the chelicerates include one group called Xiphosaura, which is the horseshoe crabs, and then the arachnids. So this is showing some subdivision among the arachnids, but when you think about those, there are spiders, scorpions, mites, and ticks. So they're all chelicerates, and we'll see why they're defined as those in a minute. Then we have this other group over here, right? They're called the mandibulates. In the arthropods, a pair of sort of side-to-side action biting jaws are the mandibles. So the mandibulates have, as an ancestral characteristic, these biting jaws. There's two main groups within the mandibulates. There's the myriapods, and the myriapods have this body plan that looks pretty similar to our ancestral arthropod. They just have a whole bunch of segments that all look the same. And we'll look at those in more detail. So the other taxonomic group besides the myriapods is pancrustacea. Now pancrustacea actually includes the hexapods, and the hexapods includes the insects. So we have a variety of sort of crustacean-like taxa that are grouped together with the hexapods. And so we call that taxon pancrustacea because if we just called it crustacea, then we have more diversity in there. So this has been a little reorganized, and there's still folks that don't completely agree on these taxonomic characteristics and how we're going to deal with these crustaceans. But I want to thank Dr. Miller for providing this diagram, because it's actually from a publication of a paper that he had published last year. So it's a very nice figure, and it's helpful for us in our class as well. All right, so we're going to look at the different groups, the chelicerates, the myriapods, the hexapods. And one of the things that we're going to look at is how the overall body is sort of partitioned. And we're going to see that all of them have a head, and the head is going to be made of a certain number of segments that have been fused together over evolutionary time, usually 5. And then in the myriapods, we're going to see this body where they have these repeating units that are all more or less the same. So basically they just have a head and body, or a head and a trunk. We're going to have one group, the insects, that has 3 distinct body sections, body regions: the head, the thorax, and the abdomen. And we'll see that an important characteristic is that they have 3 pairs of legs, and that they're all on the thorax, right? They don't have any legs on their head. They don't have any legs on their abdomen. And then another way that the body can be organized overall is to have some kind of fusion or lack of distinct separation between the head and the thorax. If that's true, it's called a cephalothorax. So the head and the thorax are sort of smooshed together. And we see this in crustaceans, and we see, so this is a crustacean. And also we see this cephalothorax plus abdomen model in the chelicerates, right? So the ones that start with a C have the cephalothorax. So this is how overall the body segments are sort of organized. And then we'll see that when we look at those, there's other characteristics that are going to help us tell the difference between our groups. So there's a lot on this slide, but we're going to review it as we go through the rest of it. So using the same color scheme that was on the tree diagram, our chelicerates include those 2 groups, the Ziphosura and the arachnids. Remember that the arachnids include spiders, scorpions, ticks, and mites. Notice that all of the chelicerates have their bodies divided into the cephalothorax and abdomen, and that they have 6 major pairs of appendages on the cephalothorax. Right? They may have other appendages on the abdomen, but they aren't going to be sticking out to the sides, and that's only in the Ziphosura. So in the arachnids, they're going to have no appendages on the abdomen. And when I say legs, usually that's something that they're going to be using for walking. So when we look at Ziphosura, we're going to see that those 6 major pairs of appendages break down into a pair with a bigger claw at the tip called chelicerae, and then 5 pairs that they walk with. However, in the arachnids, we're going to see that they have the chelicerae, which have been modified into a biting or stinging jaw, pedipalps, which are sensory organs, and then the 4 pairs of walking legs. So when we look at a cartoon of our Xiphosura, which is the horseshoe crab, the front of its body is going to be horseshoe shaped. And it's going to have the abdomen, and it's going to have a long thin tail sticking out, and it's going to have a compound eye on each side. They look like some kind of armored tank or personnel vehicle, and you're only going to see those appendages if you turn them over. But we can see that the body's divided into the cephalothorax, the abdomen, and then this tail, and in arthropods, the tail is called a tulsin. Now in the arachnids, we're going to see the abdomen, it might be quite large, and then the cephalothorax, we're going to see those 4 pairs of large walking legs. And then we're going to see the pedipalps and the chelicerae. They may have up to 4 pairs of eyes. I'm not going to be able to fit them all in. All right, a bunch of eyes, more than 2. So Xiphosura has 1 pair of compound eyes. Those are large that they you can see very easily, and then they're actually going to have another pair of simple eyes right at the nose. It looks like nostrils, but they're simple eyes. All right, so when we're looking at the Myriapoda, we're going to see that they have the little head, and then they have the long trunk with all the basic parts looking the same. They only have a simple eye, a pair of simple eyes. They've lost the compound, and they are going to have antennae, right? So none of the chelicerates have the antennae. We talk about the body sections of the myriapods. They basically have the head and then everything else. So we'll call that the body. When we look at the crustaceans, they're going to have that cephalothorax and abdomen, like we talked about before. And when we look at their body, we're going to see the cephalothorax is typically larger, the abdomen is smaller, they may have a little tulsin sticking out. And they're not going to have a small set number of appendages on particular segments. They're going to have all kinds of legs all over the place. So if we see a bunch of appendages on the abdomen, that's our best clue that we're looking at a crustacean. The other thing that's going to be usually obvious on crustaceans is that they have antennae, and they not only have a pair of antennae, they have 2 pairs of antennae. Sometimes it looks like they have a whole forest of antennae sticking out because they can also branch again. And then they have a compound eye, and there's something unique about it compared to the other groups, and that is it sits on top of a stalk. So it isn't just a compound eye, it's sort of movable on this little, it's on the tip of this stalk. So they have cephalothorax and abdomen, they have 2 pairs of antennae, they have a stalked compound eye, and then they have many legs on the cephalothorax and many legs on the abdomen. The insects are really easy. They are just going to have a separate head, thorax, and abdomen. Usually we can see that. They're going to have 1 pair of antennae, and they're going to have a compound eye that's basically flush to the body surface. And they're going to have 3 pairs of legs, and they can have up to 2 pairs of wings. So I only drew 1 pair of wings on my little insect. They can have up to 2 pairs of wings, although some don't have wings, right? So if you see an organism that's an arthropod and it has wings, it's an insect. It's got 3 pairs of legs on its thorax, and it doesn't have any other pairs of legs anywhere else. It's an insect, right? But because it doesn't have wings, that doesn't necessarily rule it out, because ants, for example, don't have wings for most of the time during their adult life. They only grow them a brief, short period of time when they're undergoing reproduction. All right, we're going to look at some pictures of these different organisms, and I want you to just bring your notice back to looking for these characteristics. So on this picture, you can see the compound eye, these simple eyes. All of our shoe crabs basically look the same. There's a handful of species and they occur in the US in the Northeast. So if you've never seen one of these things and you're like, what the heck is it? If you've never lived in the Northeast. Now, I lived in Massachusetts for a while and I still never saw one, so it's no guarantee. They also occur on in northeastern Asia, so along the coasts of Japan and Korea, you might also be able to see these on the shore. If we look on their underneath side, we'll see that they have the chelicerae, the first appendages here that have little pincers. Those are for putting food into the mouth. And then they have these 5 pairs of walking legs, 1, 2, 3, 4, 5. All right, so 6 total pairs of major appendages on the cephalothorax. This is a line where the abdomen is separated from the cephalothorax, and there's a hinge here. So the arachnids, the groups include spiders, ticks, mites, and scorpions, and we're going to be able to see again that they have this 6 pairs of major appendages. Now we're only really usually going to be able to count, obviously, the 4 pairs of walking legs. So we have the 4 pairs of walking legs, and then in scorpions, the pedipalps are modified into chela. We can see the pedipalps for this black widow spider, and then we really can't see the chelicerae in any of these pictures. And you can just see the pedipalps. All right, we know they're there though. That's what makes them a chelicerae, is those little chelicerae. So you're going to look in arachnids, you're going to look for those 4 pairs of walking legs, but you also need to know that those other 2, the pedipalps and the chelicerae, are there as well. All right. We're going to focus on scorpions and spiders in our class because the ticks and mites are very small and we're just not going to be able to see them with the naked eye. All right, in our myriapods, they come in 2 kinds. We have 2 flavors. We have chelepods, chelepoda, which are centipedes. And if we look at them from the top, they're fairly flat. They're going to have some fairly obvious antennae. They're going to have this little claw at the front. And then the rest of their body, the segments are pretty big. And they have some little things sticking out their tail. And the legs basically go straight out to the side. So we're going to see when we look at it from the top, it kind of looks flat. And that there's a good deal of space in between the pairs of legs. The diplopoda, the millipedes, are going to look different. So this one's curled up on its side, and you can see that there isn't any space in between the legs, and it turns out that they actually have 2 pairs of legs per body segment. And so it's going to make it look like they have lots more legs. They're not going to have obvious antennae. They do have antennae, but they're little, so you may not be able to see them too easily. And their little foreheads are rounded. When we look at them from the front, they're going to be more dome-shaped or rounded. And we'll see all the legs sticking out. All right, so they have a much more rounded overall body profile, where centipedes are more flat. When we look at the side, the legs actually are going to be partially curling under, whereas centipedes, they're sticking out straight to the side. So they're pretty easy to tell apart. There is a group of millipedes that are called flat-backed, right? So they don't look quite as domey. They don't look like they're so rounded, but you can still see the legs. You can still see that there's 2 pairs of legs per body segment. All right, crustaceans. So remember, crustaceans are going to have the stalked compound eye. They're going to have the 2 pairs of antennae. In some organisms, they're very obvious and large, and others not quite so much. but these are going to be almost all marine and aquatic, except for our little friends, the pill bugs, which are the only group of terrestrial crustaceans. The crustaceans are going to include things like barnacles, which we're going to have some examples of in lab, crabs and lobsters that you're maybe familiar with. In marine biology, when we're looking out in the open ocean, we're going to see copepods, copepods, copepods. They are the grazers of the sea. And our example copepod there is pregnant. These are egg sacs. So she's got these dangling off of her. Krill are a shrimp-like organism. They're also crustaceans. Our little friend, the water flea or cladoceran, also has eggs that she's carrying underneath her carapace. And then we have deep sea isopod. That's a very large cousin to the roly-poly. You can see that it has lots of legs. In fact, it has 7 pairs, so it's clearly not an insect. And it can't be chelicerate because we see that it doesn't have kind of those kinds of chela at the tip of its legs there. All right, so there's incredible diversity of aquatic and marine crustaceans. There's a variety of ones that is obvious they're crustaceans. Some of them, like the barnacles, you need to recognize what it is and say, oh, that thing is a barnacle, and then remember that it's a crustacean. So here we see this barnacle. We see all these legs sticking out from the top of it when it's feeding. And then these ones are showing they can close a little trapdoor on the top of their shell and protect themselves from drying out if they live on a rock that is where the water from the tide is going up and down. When they are dried out, or when they're not under the water anymore, they'll close this trapdoor at the top of their house, and there's a little crustacean animal living inside there. So insects, we only have a couple examples here: the ant, the fly, the beetle. You should be fairly familiar with a variety of insects already. And insects are the most diverse of all of the different groups. So crustaceans basically have taken over all the marine and aquatic environments, whereas insects have taken over all the terrestrial environments. And we'll return to insects again and look at more of their diversity in the next section of the lecture. All right, so this was an introduction to the different groups among the arthropods, and now we're going to sort of do part 2 of our lab, which is going to be diving into more detail on the 2 more diverse groups, the crustaceans and the insects. So here we have a tree diagram that's showing just the mandibulata. It's not showing the chelicerates. And here within Pancrustacea, we have a small subgrouping that's going to include all the insects here. Then we have all these other different kinds of crustaceans along here, and they have interesting names like the copepods, malacostracans, branchiopods. And we also have some hexapods that are non-insect, like our little collembilids, like there's a picture of. So it has the 3 pairs of legs, but they never grow wings. And so they're hexapods, but they're not insects. So I just want to introduce you to the idea that there's a lot more variety in these taxa than what we're covering in lab. All right, so just a reminder that Pancrustacea includes both the crustaceans and the insects, including some other taxa we aren't going to talk about. All right, so part of our examination of the insects and the crustacean in more detail are the dissections of a grasshopper and of a crayfish. So we're going to be able to look at those log branch characteristics in the dissection and also get a good look at the external anatomy as well. So there's a compound eye, there's the antennae, you can see the body segmentation when we're zoomed in on these animals, you can see the head, thorax, and the abdomen. And then when we cut into the exoskeleton and reveal the internal structures, we're going to see a couple things here. One of them is on the outside of the gut. And so when I said I promised you their excretory organ, has a great name, the Malpighian tubules. All right, so the Malpighian tubules is the name of the special excretory organ you find in the insects. Notice that they have this a complete gut shown here as food is going to move through. And these yellow structures are these digestive glands or little extensions of the gut that go out to the sides that food can stay in longer and get digested. So those are very obvious. You'll be able to see those. One of the things to notice when we're looking at the grasshopper dissection right next to the crayfish dissection is how similar their bodies actually look. Now you can look at these and you can say, oh, well, you know, this is clearly a crustacean, the other one's clearly an insect. But when we think about all the other phyla that we've looked at, the porifera, right, definitely the crayfish and the grasshopper look very much alike one another compared to a sponge. They look very much alike one another compared to flatworms, to roundworms, right, to echinoderms. They're clearly in the same phylum. They have the exoskeleton, they have the body segmentation. We even look more carefully at their structures, such as the compound eye. If we look down at all the legs, they may have little different kinds of bristles sticking out. We can find these shared characteristics. All right, our crayfish is in a group of crustaceans called the decapods, and it gets that name because it has 5 pairs of walking legs. It has antennae, it has a stalk compound eye, and in the dissection you'll be able to see quite clearly that it has the same basic internal body organs, this complete digestive tract, it has the small brain and the ventral nerve cord. And what's being shown on this figure as well, though it's very hard to find in the dissection, is the green gland. And the green gland is what the excretory organ is called in crustaceans. And so we have the Malpighian tubules in insects, we have the green gland in crustaceans. Another way that we're going to distinguish between crustaceans and insects is how they go through their development. So when they are growing from an egg through an embryo into a juvenile, they're going to have to molt in order to grow. And so often we're going to see, just like in the nematode, that we have a series of larval stages, and that as it's molting, then we're going to maybe be able to distinguish between those different stages. So each of these different stages, if it can have multiple ones as it goes through development, are called instars. So we might say, instead of a larva 1, 2, 3, or 4, we'd say instar 1, 2, 3, 4. Now the crustaceans have really interesting and varied stages of larvae that they go through. The first one is usually called nauplii. And then, for example, a crab goes from being a nauplius to being something called a zoea, and then something called a megalope. The reason they have different names is because they look really different from one another. So every time it goes through a molt, it can have the opportunity to sort of change its morphology, right? So crustaceans are going to gradually go from one kind of morphology to the next as they're growing. You're thinking to yourself, wait a sec, what about a caterpillar turning rapidly in one step into a butterfly? That's a special version of a life cycle that we only find in some insects. So in insects, we're going to see that there's 3 versions of the life cycle of how do they grow, how do they get from being a larva to being an adult. Okay, so here's an example of the crustaceans. And we start with the nauplius down here. And as it's growing, you see it has some different sizes and proportions, and then it's eventually going to grow into a full adult. So at each molt, it can change a little bit its proportions or its appearance. When we look at all of the different crustaceans in a little more detail, some of the ones that we're likely to see in the intertidal zone here in San Diego is an example of amphipods. You might know these as beach hoppers, and they're going to be flattened from side to side. In fact, they're so flat from side to side, sometimes if you see them and they're on a rock, they're actually going to be tipped on their side, and they're going to be doing this kind of Pilates thing where they're squishing, they're trying to turn themselves up, but they can't really sit on their legs. They're sort of so curved around. Isopods are going to be both marine and terrestrial, so are rock lice and wood lice. Decapods includes crabs, lobsters, and shrimp. Copepods are things that we call copepods. And brachiopods is the group that's going to include brine shrimp and our water fleas like the cladoceran. Here's an example of a fairy shrimp. Again, I have a pregnant female. She has an egg sac that she's carrying around there. They swim around on their backs, so she is in fact in her appropriate orientation. And then the barnacles are in a group called the Cirripedia, or whisker or feather-footed. So this is, again, it's going to be this volcano-shaped animal that has a trap door at the top, and then these legs are going to be sticking out when it's time for it, when it's underwater and it can open up, it's going to sweep its legs through the water. All right, so there's some crustacean diversity. Now we're getting back to the 3 insect life cycles. So when we think about that transition from a caterpillar to a butterfly, that's where we have a complete metamorphosis, That in one step, we go completely transforming from the larva into the adult. They don't look anything like one another. So when we have complete metamorphosis, we're going to call that larva something, right? When it's a butterfly, we're going to call it a caterpillar. When it's a beetle, like this flower beetle, we're going to call it a grub. You find these in our garden sometimes when you're digging in the ground. When we're looking at flies, the larva of a fly, we usually call it a maggot, and we don't really want them around anywhere, but they're going to turn into a fly when they're finished developing after the pupa. So not all insects have this though, right? So this complete metamorphosis is shown over here. We have an example of a moth. It's going to grow as a caterpillar for a while, and when it gets big enough, it's going to go through a pupal stage and then become an adult. So we'll call this one a complete metamorphosis. So holometabolous life cycle. We also have some insects, the most primitive of insects, similar to this guy. It's called a silverfish. You find these in boxes in our garage, sometimes in books or in drawers or in your bathtub. They never have any wings, right? So they're never going to grow wings. And the thing that distinguishes an adult from a larva is that the adult has wings, fully functioning wings that fly, right? So the caterpillar doesn't have wings, the butterfly does have wings. Ametabolous, there's not very many groups. They have no metamorphosis, they have direct development. And they have no wings, they never grow any. So the most primitive insects, when they first came onto land, compared to the marine organisms that they derive from, some crustacean-like organism, they didn't start having wings right away. Hemimetabolous life cycle, there's quite a few taxa that have this. I didn't complete, ironically, I didn't complete my word complete. Hemimetabolous is not a complete metamorphosis. It's an incomplete or it's gradual, right? So we have each life stage is going to look more or less like the adult. But what happens is the wings grow little by little. And these stages aren't called larvae. They're called nymphs. And the nymph, when it's very teeny, isn't going to have any wings at all. And the wings are basically going to grow as little nubs. So we can't see it very well on this diagram, but they'll have a little stubby wing. And then after another couple molts, they'll have a bigger wing. And then the wings keep getting bigger as it goes through all these subsequent molts until the time that it's got its fully sized wings here. They overlap one another, so they're hard to draw. All right, so we'll look at some examples in lab of which kinds of insects have which kind of life cycle. But the complete life cycle is the one that is for the most kinds of common insects, the ones that we the most, you know, if you ask your little cousin what are some different kinds of insects, they'd say, you know, a butterfly, a fly, a honeybee, an ant, and those are all different ones that have this holometabolous life cycle. All right, we're going to quickly run through some pictures, and this is diversity within the taxa of the insects, and so usually these are called orders. So at the order level, we have this taxonomic group which has the silverfish in it, and it's had a recent name change. So depending on how old the document is you're looking at, it could be called Zygentoma, or it could be called Thysanura. And they never have any wings, and they have these long, thin antennae, and they have these tail bristles. Oops. These tail bristles, which you can see here, which are called circi. Right, and the antennae are proportionally very long. Odonata are the dragonflies and damselflies. The antennae are so teeny you can't see them, so they're there, but you can't see them with your naked eye typically. And they're going to have two pairs of translucent wings. All right, so what I mean by translucent is that you can look right through them. So if we look at this dragonfly over here, they're clear. Dragonflies are going to hold them out to the sides. Damselflies, when they're not flying, are going to fold them up over their backs. So sometimes it's hard to tell on the damselflies, but they have this long stick-like body, very large head, very large eyes. So you can probably recognize those two. Coleoptera are the beetles, right? Beetles. And what we're going to notice about beetles is that whether or not you can't see the wings usually if they're sitting still, because their wings go under a hard protective coating. And what we're going to see when we look at beetles is that they have their head is usually fairly set off from the rest of the body, and that they have a straight line where those two hard coverings come together. So they're called elytra. That doesn't matter so much, but we see that they have this T that you can notice across the top of their back. And these are small, so some of them you can't see very well. You can't always see it, but that's a good clue to notice that you're looking at a beetle. Now, they may suddenly surprise you by opening those elytra up and flying away, because their wings are hidden underneath them, like a ladybug. Hymenoptera are ants, bees, and wasps. And one thing to notice about them is that they have this real skinny waist, or the connection between their thorax and abdomen. So when we see this little skinny waist, then normally we notice it's Hymenoptera. And we may notice one, or they can have no wings if they're in certain parts of their life cycle, like ants are most of the time. And when we see a bee or a wasp flying, or a fly flying around, sorry, not a fly, that we may only see one pair of wings, because the hind wings are smaller. Right. Try and recognize that it's an ant, a bee, or a wasp, and you know it's a Hymenoptera. Lepidoptera is the butterflies and moths, and they have two pairs of wings, although again, they're fastened together, so sometimes it's hard to tell that they're two. And that if we zoom in on their wings, we see that they're covered in tiny scales. The difference between a butterfly and a moth isn't just that one is awake at the daytime and the other is awake at night. Moths tend to form sort of a triangle shape when they're perched, and they hold their wings down flat. Butterflies, typically, when they're perched, you're going to see them holding their wings up from the side. Not all butterflies are brightly colored. Not all moths are drab and dull. But if you're seeing them perched or sitting, then you can usually tell which is which. Lepidoptera. Orthoptera is a group that includes grasshoppers, katydids, and crickets. And when we look at their legs, we're going to see this super huge, like, bodybuilder thigh of their leg that's modified to have lots of muscle in it and for jumping. Now they do have two pairs of wings, but usually we aren't going to see them. So they're going to be folded up when they're sitting around. Occasionally, again, they'll jump and then they'll fly off and they'll really startle you. So this picture is showing the wings out. Normally we're not going to see them like that. We're going to see them looking like this grasshopper here, and we're going to think they don't have any wings at all. So even though they do have wings, when we look at our key to the insects that I'm going to give you to try and work on learning to tell the difference between them, we may not see the wings. Right? So the questions I'm asking you to try and help you identify the insects is going to rely on what is it you're looking at. So you're not going to have to know, for example, looking at a grasshopper that it has two pairs of wings. You're just going to be able to look at it and see based on what you see. The diptera are flies, mosquitoes, and midges, which are sort of little things that are really teeny and tend to swarm around and be very irritating. They have one pair of translucent wings that you can see. Flies are a very diverse group as well, but they're fairly small, so you may not notice the diversity among them. Hemiptera are the true bugs, and true bugs have their wings crossed with one another, and so when we look at them, we see, again, like beetles, we may see that they have kind of a shape straight across their back, but then their wings are crossed to one another, and so we'll see an X shape. If we look at their face from the side, they also have their mouth is a proboscis, or like a straw, and it's going to be sticking down underneath their face. So you can't see it from the front, but if you have a large enough one and you turn it on its side, they'll have this little snoot that's curled underneath the front of its face. And finally, the Dermaptera are called the earwigs. Now, the easiest thing to notice and the scariest looking thing about them is that these big circes. So we have the little pointy circe sticking out from the back end of our silverfishes. The one for the earwigs is an actual pincher, and it can actually grab things with that pincher. They do have wings, but again, they're mostly going to be folded up, and they're covered with these sort of leathery coatings. So typically you aren't going to see or notice the wings on earwigs, but they all have that big pincher at the back end. The Blattodea. Blattoids, now the most common thing that we're going to see among this group is the cockroaches, right? The cockroaches have a flattened body. They have wings, but again, they're going to be folded up and you can't see them, and they're going to have long antennae. All right, so this has been a very quick and brief tour through a small fraction of the diversity of insects. These are the orders that it's fairly common to see in our everyday lives. I think I've seen almost all of these groups around my house in the last couple of weeks. I even have the occasional cockroach in my house, or in the yard, right? So these are fairly large so that you'll be able to see them. And so we're going to have some examples of these to look at and practice telling the difference between. All right, so for two phyla that are fairly diverse, and the arthropods are incredibly diverse, we have had just a very brief introduction. And if you're excited about arthropods, in particular insects, the field of study of them is called entomology. So you can take an upper division entomology course and learn about all the other groups and build your own insect collection. And so it's a really interesting field of study. The very last couple slides here, Dr. Miller stuck these in case you're super interested in the taxonomy of arthropod groups. So if you want to pore over these diagrams and look at some of these different kinds of species and look and see the branches on the tree and see how they're related to one another, these are for your interest. And here's another one, even more complicated looking one. This one zooms in on the insects in a lot more detail. All right, so there's a couple dozen of different groups within the insects. All right, that is our lecture for today.